Good evening. I Atin Chatterjee, research student of IISER Kolkata. I am going to show a presentation on the topic of Catechol Oxidase Enzyme. We see brown spot appears on the fruit. If we cut a piece of an apple and keep it in air, it becomes brown after some time. The event is called as the fruit browning. People with chemistry background always try to investigate such biological phenomenon in terms of inorganic redox reactions. Actually, molecular oxygen uptake by catechol oxidase enzyme takes place, and these plays the crucial role herein. Next, catechol monophenol or diphenol oxidizes with molecular oxygen directly and from the quinone corresponding ketophrome, which undergo polymerization to form the melanin, a blackish brown color pigment, which is responsible for the event fruit browning. In this case, monophenolase is tyrosinase and diphenolase is catecholase. And what is cressolase activity? Cressol to corresponding quinone derivative is actually the cressolase activity. Now, active site. The enzyme first isolated from the potatoes in 1937. It contains the type 3 copper proteins. Type 3 copper proteins is actually the dinuclear copper core bridged by intervening ligand like H2O, hydroxo or oxo, etc. The copper 2 surrounded by the nitrogen atom of histidine amino acid residues and adopt the trigonal bipyramidal geometry, bridging ligand, in this case OH-. Copper 2 centers are 2.9 angstrom apart from each other and antiferromagnetically coupled through the intervening ligand OH, and it produces the EPR silent signal in this enzyme. More details: active site and binding site. Figure A, blue color is helix, yellow color disulfide bridges, orange color beta strand, and figure B represents the actual coordination sphere and geometry around the copper center. Figure C, phenylthiourea inhibitor binds, in which sulfur atom of the phenylthiourea replaces the bridging ligand OH and conformational changes occur. Figure D, oxygen binding site is represented in green color. Now mechanism. Two mechanisms proposed. First, Krebs mechanism and another Sigben mechanism. Both mechanisms suggested phenol is converted to the corresponding quinone derivative, but one difference therein: Krebs mechanism starts with the oxyfrom of copper core that is made from actually, whereas the Sigben mechanism starts with the deoxyfrom of the dinuclear copper core. First, we explain the Krebs mechanism. Oxyfrom of dinuclear copper core. In this enzyme, catechol binds in monodentate fashion to one copper center. Then enzyme gets reduced to copper 1 and quinone is formed. Then second catechol binds similar monodentate fashion and molecular oxygen binds as hapto-2 coordination mode. It is actually the symmetrical binding. Quinone is formed through such subsequent electron transfer or redox processes. Sigben mechanism initially stated starts with the deoxyfrom and catechol binds and transfer the proton to bridging OH and hydrogen bonding interaction occurs. Oxygen displaces the bound H2O and superoxide radical anion is formed. In this case, mixed valence copper 2 copper 1 core is formed. Superoxide abstracts the proton and electron from the bound substrate, in this case catechol. To release the quinone, an electron is transferred from the semiquinone radical to copper 2, leading copper 1 state. ABC step is explained easily. Now, cleavage of OO peroxo linkage by transfer of two protons from substrate and electrons to the peroxides occurs. These steps is explained in D and E. Another electron transfer occurs from semiquinone radical to copper 2 center that leads the restoration of the initial hydroxo bridge. This is the pictorial representation for the catalytic cycle of catechol oxidase. Actually, catechol gets converted to the corresponding quinone derivative through various electron and proton transfer processes. Our focus. So, why we learn such interesting enzyme? If we use the molecular oxygen directly for oxidation of various organic substrates, we use such enzyme as alternative to the standard oxidizing agent potassium dichromate or the potassium permanganate because it is environmentally benign and economically viable pathway. A funny speech is that if we wish to eat and buy some good quality of fruit, we must quantify the patholage or polyphenol oxidase or catechol oxidase enzyme in it, because the greater is the possibility of the melanin formation, the greater will be the possibility to save the fruits from insects or pathogens attack. So before buying fruit, we must quantify the polyphenol oxidase enzyme in it. Besides it, this melanin and the enzyme have been paid the enormous attention for the therapeutic purpose or hormonal therapies like the adrenaline, vasopressin, and or catecholamines or dopamines, etc. etc. Thanks a lot.